We're live.
Hello everybody. Greetings from Heart Care Foundation of India. I am Dr. K.K. Agarwal, President, Confederation of Medical Association of Asia and Oceania, past National President, Indian Medical Association, and Heart Care Foundation of India President. Welcomes you all on 40th weekly CMAAO meeting. I'll request SM Sajjad Saab to chair this meeting. Dr. Kasir Sajjad to chair this meeting. And then once the official chair comes, both of them will chair this meeting together. And the topic we are going to take about is the new corona strain in various countries. Does it matter? Is it a matter of concern? With that, we'll request Dr. Sajjad to give introductory remarks. Dr. Sajjad.
Thank you very much, Mr. President. Actually, I'm sorry that I missed two CMAAO meeting because I'm suffering from COVID.
You are suffering from COVID. Oh my God.
Today I'm much better. But little weakness. I feel little weakness and sometimes coughing. So this is very. I pass a very horrible time, but thanks God I am much better now.
हम सब लोग पहले आपके लिए दुआ करते हैं और प्रेयर करते हैं कि आपकी वीकनेस जल्दी से जल्दी खत्म हो जाए. We pray.
Thank you very much.
Pray to the Allah and God for your recovery.
Thank you very much, Mr. President. I hope today meeting will, as always, will be very informative and very fruitful. Just go ahead, Mr. President. Thank you very much.
So thank you, Chair, for starting this meeting. I hope more people will join. So I'll give a presentation and try to be as basic as possible. Now this presentation is not possible without acknowledging Dr. Angelique Kordze, the chairperson of South African Medical Association. She's a part of our CMAAO group. Then Dr. Pavitra Venkata Gopalan. She is the first PhD on coronavirus in India. Dr. Navedita Gupta, who is the head of COVID-19 in India in ICMR. Dr. Jayanti Shastri, the first one to report official genomics of reinfection in India. Dr. Anita Chakravarti, a microbiologist retired from Maulana Azad Medical College. So these are the inputs which I have interacted with them over a period of time in my various shows. Some of them I've used their slides. So instead of acknowledging each slide, I'm acknowledging all of them together, and I have made my presentation. My first slide goes back to a pattern. We have a pattern of illnesses. And if you see a pattern of illnesses, if you see UK, the second peak is much bigger than the first peak. So the second peak is much bigger than the first peak. So all the countries which are showing the second peak bigger than the first peak. So whenever there is a second peak means two things: laxation of the behavior of the public with available naive population or the second thing will come is mutation of a virus. Now, why should a second peak be larger than the first peak when everybody has learned the prevention? So the common sense is whenever the second peak is larger than the first peak, first look for a mutation. And that's what now we are observing in South Africa and in England. We are seeing the second peak larger than the first peak, and there has been a mutation of the virus. So I am pretty sure all the countries which are showing a second peak larger than the first peak, the virus have undergone a mutation. So what is the definition? The term variant, strain, and lineage are uniformly used. The term variant, strain, and lineage are synonymous in scientific terms. But for when we talk to the public, we talk about the word mutation. Okay. Now, what is the definition? There are whenever a new variant is there, we call them as VUI. VUI means variant under investigation. And VOC means variant of concern. So variant of concern and variant under consideration, under investigation. So all these viruses were under VUI. Once they have been confirmed to be of concern, they will be called as VOC. Now you will find in the media, the UK virus has been changed from VUI to VOC. That means investigations are complete. Now this is a variant of concern. Now, RNA viruses are likely to replicate. Why? Because they are unstable. And if you see coronavirus is less likely to replicate than HIV virus because of the size of the virus. More the size of the virus is bigger, more will be, and depending upon, more will be chances of mutation. That's how the thing will come. Now let's see what is a mutation. There is a name called Amita. You, instead of one I, you add two I. She will still be called as Amita. If you take the I out, still from a distance, you will say Amita. If you add two A's, still you call it as Amita. If you make the A smaller, even then you will call it as Amita. But if you change I to U, she will become Amuta or Umita. Now you know that the characteristics of a virus has changed. Therefore, minor mutations will not make a difference in the behavior of a virus. And that is called antigenic drift. When there is a major change in the virus, it changes its behavior. It is called antigenic shift. Now, if you see the difference between a DNA and RNA, there are only four, cytosine, guanine, adenine, and uracil. In permutation combinations, they make everything together. So the nucleotides are only cytosine, guanine, adenine, and uracil. In DNA, uracil replaces with thymine. So only four base blocks make the whole RNA. RNA makes the whole protein. So these are only CGAU. These, now don't see the upper one, which is a DNA. Look at the RNA level. At RNA level, three of them together in combination, CGAU, three of them in combination will make the amino acid. So when AAG unites together, they will always make, three of them will make an amino acid. So AAG means lysine. AAA means also lysine when there is a mutation. So if there is a mutation of AAG or AAA, it will, both the body will recognize it as lysine. Whenever there is a UAG, means UAG, this means the protein formation will stop. This is called stop codon. So if there is a mutation which ends up into a UAG mutation, that means, that would mean the protein synthesis will stop. AGG will become arginine, ACG will become threonine. So therefore, one word can make all the difference. Okay. Now, when we talk about messenger RNA, three of them will make one polypeptide. This is how the polypeptides, the changes occurs. I'm not going into details. So there are multiple mutations occurring. They will keep on occurring. Which mutation gives them the advantage will persist. So that is called as a selection pressure, survival of the fittest. So if there are hundreds of mutations, whichever mutation helps the virus will persist, rest will be deleted. Now, why should we understand genomics? How does it matter? We should only know this is virus, this medicine acts. No. There are reasons to understand genomics. Genomics help us to discover the virus. If the genomics was not there, the COVID virus, we would not have known what are the characteristics. It helps us to develop diagnostics. It helps us to develop vaccines. It helps us to track the transmission. Like you are talking about, is has it entered Pakistan? Has it entered Singapore or not? It helps us in identifying the reinfection, and it helps us in understanding the host virus interaction. What are the implications? If there is a mutation, it can change the way it spreads. It may be more spreadable in children, old age, antenatal. So when there was a 614 gene mutation, that mutation allowed it to spread more. So that is called ability to spread more quickly in human. It may also be a mutation which leads to more severe disease, more inflammation, more pro-inflammatory disease, more pneumonia, more ARDS, more cytokine crisis. Ability to evade detection by diagnostic tests. So because of this S gene mutation now, we are now speculating that the RT-PCR S gene test may not be reliable. Then decreased susceptibility to monoclonal antibodies. And then the new mutation may be in such a way that you will not get formation of antibodies at all. The cellular antibodies may not form. The humoral antibodies may not form. So these are the 6 areas of concern for any mutation. Now here comes the virus. Top one is the SARS-CoV virus. Bottom one is the MERS-CoV virus. And you will see most 67% viruses, they exchange the same shape. Now here is the SARS-CoV virus, that is COVID-19 virus. So this is the genomics of the COVID-19 virus. Now it is important to understand that it has a spike protein. On one side of a spike protein, there is ORF1A and ORF1B. These are non-structural proteins. And then you have ORF 3A, 3B, 7A, 7A, 8A, 8B, 9B. And then you have structures called spike, envelope, membrane, nucleocapsid. Now we come back to the S because that is a matter of interest to us. This S is made up of 1,255 genes. See here, there is one gene and 1,255 genes. Out of these, various genes are there. It has divided into two, S1 and S2. In S1, there is RBD. This is the area which enters the ACE2 receptors and binds it. And under the RBD, there is RBM, which is the actual part of the RBD which locks with the lock of ACE2. So RBD is receptor binding domain. RBM is receptor binding moiety. Now, whenever you see a mutation, you start looking for, is it in ORF1? Is it ORF1B? Is it in the spike? Is it in any of these areas? So out of 23 mutations in UK, 8 mutations are in the S protein. Now you are concerned. The rest you can ignore. But if there are mutations in the S portion, you are worried. And when you say okay in S1, so let me see, is it in S1 or S2? You say S2, you say okay. But if you say, oh my God, it is in S1, I'm worried. When you have it in S1, you want to know, is it in RBD or non-RBD part? You say non-RBD, we say okay. But if you say, oh my God, it is in the RBD part, I'm worried. Then you say, in the RBD, is it in RBM? Oh my God, you say, I'm more worried. This is how you decide, theoretically, which mutation will cause what. Now, the South African mutation has 3 mutations in RBD, one here, one in the red, one in another green. The UK mutation 501 is in C. It is after RBM. It is in the green part. Theoretically, South African variant is more contagious, more antigenic than the UK one. Theoretically, we are not sure because it is one of them is in RBM. In UK, one of them is 501. That means between after the RB moiety in this green portion, rest of them are in other areas of the spike protein. So therefore, in every mutation, one mutation becomes a mutation of concern. For that, we must know what are the roles of various genes in the body. So ORF1AB to 16, this is 16. These are all non-structural proteins. They cause virus replication, proofreading, suppresses cellular immunity, and unknown. For this protein, proofreading is NSP14. So NSP14 is the most important. NSP14 is the most important, which is responsible for proofreading, which we will come later. Spike, ORF3 is for virus release, envelope is for virus assembly, membrane is from the virus envelope, then these ORF6, 7A. So you can see the mutation in which can cause what in terms of virus, but nobody is talking about the virulence. They are all talking about the viral spread in the body. Now, how do you classify various mutations? China mutation started with L, then became S, then V, then G, then GH, then GV, and O is unclassified. Currently, all the viruses, new viruses are under unclassified. Now, how do we decide? So in S clade, it is 37th gene, L replaced by F. So therefore, in 37 number gene, that means in this blue portion, there is a replacement of L with F. We will discuss this, what is L, F. These are the name of the amino acids. I'll give you a graph to decide. Then came V clade. That means on 251st gene, G becomes V. Glycine becomes valine. Then came G clade. This is called D614G. Aspartic acid became glycine at 614th location. Then becomes GH. That means there are two important mutations. One is D614G. Apart from that, in the NS3 ORF3A, in this GH, now we again come back here, ORF3A is responsible for virus release. So therefore, GH mutation will increase the viral release. Then we have GR and GV. And if we see in India, similarly, every country will have their mutations. So we can see out of 700 sequencing done, out of 2,882 sequencing done, this is what we see. 1045 GR, GH, GS, LV, and you can see L and V are practically absent. So every country will have, so in India we have 2,882 different types of mutations. Can you understand? So we have out of this 460 are O unclassified, and so more than 500 different variants are there. So mutations are going to be there. Mutations have no value. So when mutation becomes of concern, then it becomes important. And you can see how many different types of distributions of clades are there in India. So being a RNA virus, it is likely to mutate. It is likely to mutate. Now here is a slide which tells you the difference between influenza virus and SARS-CoV-2 virus. And only one thing is, in influenza virus, the proofreading NSP14 is not there. And in SARS-CoV-2, proofreading NSP14 is there. So when there is no proofreading, there will be more mutations. And when you have a less, and if you have a proofreading protein, you will have less mutations. That's the reason CoV-2 has less mutations compared to influenza, because of presence or absence of the proofreading mechanism. Now, these are different mutations so far. This is only, and if you can see here, these are the mutations which are already present in the society. So you will see 6970 is already there, and 501 is already there. So most of the mutation K417 is already there. So these mutations are already there in various sections of the society. So we come back to the first mutation of importance called D614G mutation, which is the present mutation in the whole country. Everywhere we have D614G. It is the same mutation when there was a big news that an Indian passenger through Bhutan came to Malaysia and caused this. And now universally throughout the world, we have D614G mutation. That means 614G means this is in non-RBD part of the virus. Because it is in the non-RBD part of the virus, it was responsible for two things. Coronavirus replication, it increased its infectivity, but it also made the virus more vulnerable to neutralization by antibodies. Therefore, this virus was related to reduced mortality, increased contagiousness. So having D614G is beneficial because it increased the infectivity, but also made the germ more vulnerable to neutralization by antibodies. Now we come back to the Nigeria. There is a virus mutation change which is called 681. 681 is, now you can see this is in this part. 614 is here. It is similar to the 614. This is called in Nigeria. Now they are calling 681 mutation. That means from proline to histidine. We will talk about it in a minute. And now we come back to the UK one. The UK became from virus under investigation to virus of concern. It is classified at 2020 means the year, 12 means the month, 01 means the first mutation. As on today, it has identifiable 23 gene mutations. The main substitution is 501, that is NY, that means asparagine replaced with Y tyrosine. Then it has 3 deletions, 69, 70, and 144 deletions. Other substitutions are A, 570D, D614G, 6P681H, T716I, etc. Now see here, P681H. This is what I told you in Nigeria. So in UK, in Nigeria, you have P681H, one of the mutation which is also present in, where did we go? Which is also present in the UK strain. The UK strain has multiple mutations. One of them is in the RBD, but not the RBM. Now, if you see on the right side, you can see practically everywhere the arrows shows that there is a mutation. Therefore, it became VOC, because on multiple areas, there is a mutation. On multiple areas, there is a mutation. Now, 6970 deletion. 6970 deletion, it changes in the spike protein shape. Whenever there is a deletion, if you take your one of the arm away, there will be a change in the body shape. 501 gives stronger binding to ACE2, and Y145 deletion, it is a region which is responsible for neutralizing antibodies. So we don't know yet, but it's a matter of concern. 145 is in the first blue portion. So therefore, Y145 deletion, how will it affect the neutralizing bodies? The time will tell us. If you see these mutations, the P681H, which is also there in Nigeria, which is also there in Nairobi, it is near the S1, S2 furin cleavage site. With high variability in coronaviruses, this mutation has also emerged spontaneously multiple times in various countries. So there are other types of mutations which I have read. I'm not going into the detail because the retention power is small of all of us. You can see in UK, the second wave is stronger than the first wave, broader than the first wave, earlier than the first wave. Therefore, looks like from September to December, all these cases are new strain. Now come back to South Africa. South Africa, the main strain is 501. That means 501 is here in the blue. Then it has K4179, which is in this green. Then 484, which is again in the RBM. So it has three mutations. All three of them are in the RBD. Therefore, South African graph, let's see what happens in the South African graph. If you see here, it is the first wave. All the viruses are either green or blue. That means the strains are different. And suddenly now you see all the virus strains are yellow. So this is spreading much faster than the UK. And it has practically replacing all the strains. If you see last line, 100% cases are the new strain. 100% cases are the new strain. This is how the spike protein looks like, which has three petals. All three petals have different proteins, and this binds to the ACE2 receptor. So each of the three petals of the spike protein carries a hook called RBD. That means there are three RBD in each petal. Each RBD has a RBM. Whichever unites with the ACE2 receptors, it enters. And you can see these three mutations, N501, 417, 348, and they are ready to attach to the ACE2 receptors. Now in this graph in the South Africa, you see the second graph is earlier. If you see the space here and the space here, that means the space when the cases started, they were less than 1,000, and here it is more than 2,000 in the early phase. So it is earlier, bolder, stronger, broader, and you can see here the second wave is stronger in Western Cape, and in Nuttall Place it is earlier, broader, earlier, because it is coming earlier than the second wave in time. So therefore, these are the graph on the right side, which tells you the abbreviations. When there is a shift from one to another, these are the abbreviations. Now we come back to another mutation which has occurred in Denmark, which is important to us. In Denmark, what happened was there was a strain. There were 5 clusters were noted in Denmark. The cluster number 5 was significant because this was also occurring in the mink animal. And the specific mutation was 6970, the same mutation in UK. The same mutation in UK. Then Y453, 169, 229, there were multiple mutations. But I am now only going to say is 6970 was seen in Denmark. The Nairobi mutation was also seen in Nairobi. So multiple mutations are occurring in multiple companies. In UK, they gathered together into one virus. So multiple mutations in various companies were adopted by the UK virus and converted them into a 23 mutation virus. That is called survival of the fittest. So therefore, this Denmark, which they said the virus became extinct on November 19, 2020. When the virus became extinct, it passed on the information to the UK virus and entered into the UK virus. Now comes is the most important thing. Now I want to come about this RNA-dependent RNA polymerase, which is called NSP14. NSP is non-structural protein 14, which is responsible for photocopying. If you damage NSP14, photocopier machine becomes defective. The proofreading of the photocopier machine becomes defective. And The photocopy RNA dependent RNA polymerase means replication of the virus, which also contains proofreading. Now, what is happening is all our drugs today are working at RNA dependent RNA polymerase, remdesivir, favipiravir, and they are also blocking NSP14. That means in an immunocompetent phase, it's okay. But in an immunocompromised phase, where the virus replication is persistent but subdued, but persistent, NSP14 taking out the proofreading quality may be harmful. So this is one of the theory for multiple mutations occurring in the world is misuse of antiviral drugs after in immunocompromised patients after 7 days. It's a theory, it's not an answer. This theory yesterday was shared by Dr. Pavitra with me in my Met Talks with Dr. K.K. Agarwal. She is of the feeling that NSP14, anything which kills NSP14 in an immunocompromised patient, not in an immunocompetent patient, will cause formation of mutations. And another theory which is coming is that whenever you give convalescent plasma to immunodeficient patients and the virus remains alive, the virus is neither killed nor active. In that cases, multiple mutations can come. And they have given an example of South African variant N439K, which happened in a patient who received multiple plasma therapy where the virus survived for 105 days. So if you have an immunosuppression state of the body, multiple use of virus, multiple use of drugs which are not able to kill the virus may produce more mutations. That's what I wanted to say and share with you all. I had tried to be as basic as possible. And so over to all of you for discussions. Dr. Sajjad, you can invite discussions on this.
Yes, thank you very much, Mr. President, for very informative presentation.
Sir, may I ask you a question?
Yes, Ajay, go ahead and you can ask question, Dr. Ajay. Dr. Ajay Kumar, you can ask question, please.
Hello, am I audible?
Yes.
Yes, now.
So is it possible that the new mutated virus is so less virulent that we may like कि समझो चलो जी इससे infection हो जाए हमें तो अच्छा सबों को फैल जाए.
See, we don't
the
know at the
diseases-
moment. Ajay ji, this is an international forum. We stick to English. And the new virus, how will it behave? We don't know. Remember, from SARS-1, MERS also came, which was much more virulent. So a virus may become more virulent or less virulent is dependent. Remember that when SARS came, MERS became more virulent. Then COVID-19 became less virulent. Now the viruses are becoming less and less virulent, but you never know. But you never know. And you also should remember that the viruses can also be made in the laboratories. So you can make a more virulent, making a more virulent virus can be a virus war. Many people are saying this is that we are going through a world war, a virus intelligence versus a human intelligence. So every time you produce an antibodies, virus changes its behavior. So it's a fight between the two. So you cannot say that the virus will always go towards less virulence. Ideally, yes, but you never know.
Okay, anybody else? I have also two questions, Dr. Agarwal.
The one is that the about the vaccine. If there is a lot of mutation and what you have told, uh, the one vaccine can affect all these mutation virus.
So my see the things are viruses. The vaccine is supposed to take all the common prevalent strains which are there in the market. But
Hmm.
my question is that you remember that the spike protein vaccines, I'm not talking about the mRNA.
Yes.
The mRNA, the spike protein virus is only going to handle the spike mutations. It is not going to handle the non-spike mutations. If the normal person develops antibodies, so it is all at this moment we all are learning. We still don't have lots of answers to the questions. We still don't know. As on today, every company is saying is my vaccine will work, my vaccine will work. But the efficacy may reduce. But at least it's clear that the vaccine will reduce the severity of the illness. Even if after the vaccine, if I get the disease and does not develop the severe form of infection is okay. But we don't know why there are so many mutations, why we are worried now, why so much fuss now, is only because of the vaccine. The vaccine may not work.
Okay. My second question is concerned to me that definitely I have developed the antibodies. How much time it works? 3 months, 6 months? Do you have any idea or any confirmation?
Sorry. So Sajjad has asked a question that he has suffered from corona illness. So there are certain answers to number one. If he was asymptomatic, less antibodies. If he was symptomatic, he will develop more antibodies. If he had pneumonia, he will develop further more antibodies. So antibodies will depend upon, also will depend upon your severity of illness. That's one. Number two is two types of antibodies are formed. One is called as a cellular antibodies, and second is humoral antibodies. So we need to find out, are you developing cellular antibodies or not? So normally there is a rough way of finding out. When you take the vaccine, you do your CBC every day and see whether the lymphocytes are increasing or not to find out whether the cellular response is there or not. And then you will develop the humoral antibodies. In humoral antibodies, they will be of two types, the S antibodies and N antibodies. Because we are using the spike, so there will be only S antibodies which are formed. N antibodies are natural infection. S antibodies are the vaccine antibodies. So in your case, you will develop both the types of antibodies, N and S. So we need to check for both antibodies. Sometimes N is formed, S is not formed. So after 28 days, you should check for formation of N and S antibodies, whether they are formed or not. And then every 3 months, we check whether they are disappearing or not. In some cases, the antibodies will keep on rising. In some cases, the antibodies will keep on reducing. But remember one thing, because this vaccine requires two dosage, it means the second infection, if your first infection does not show antibodies, the second infection will produce antibodies much faster. So those people who do not have antibodies formation at all, and if they develop second infection, then their antibody formation will be very, very, very high. These are the experiences of mine.
Okay, how much precautionary measures one
So
who have suffered.
so I need to know about your mode of presentation. What was did you had loss of smell, loss of taste?
Slightly, but I have
Slightly.
a fever 101,
That's okay. That's
severe,
pneumonia. So fever more than 101 means pneumonia. CRP more than
severe.
10 means pneumonia. So what was the
Uh,
pneumonia
bo-
score?
yes, it was in the lower area,
What was the
very
score? Out
slightly.
of 25, what was the score?
This is, I don't know, but I was consulted to the pulmonologist and the physician
So loss of smell,
to
loss of taste means the person is good prognosis. They will have pneumonia. The pneumonia score will always be less than 50%. They will have involvement of pneumonia, something like 10 to 25%. Now, these are the people, because of pneumonia, they may have post-COVID cough and breathlessness because of bronchitis, which will require a treatment for 3 months. In such cases, we need to control the CRP to less than 1. And if the CRP is more than 1, we will add colchicine in such cases. And recovery is 100% in cases with partial loss of smell. Now, in Pakistan, what we are seeing is, and which is my speculation, that if in the society loss of smell and loss of taste is more than 30% or 35%, it may be a new virus. It may be a new virus. There is a second wave in Pakistan. And if you see the second wave in Pakistan, let me show you the second wave in Pakistan. What is the difference? So let me show you the second wave in Pakistan. There is something to learn from the second wave in Pakistan. I'm still trying to, yes. So PAKIS. So the second wave in Pakistan. So if I share it, now if you see this second wave in Pakistan, it is earlier, it is broader, but it is smaller than the first wave because the peak has gone and it is coming down. But if you see, it is earlier, but the peak is more because, so it's a mixed reaction. That means the second peak here, you should check for the new strain, but all the cases may not be of the new strain. That's how Pakistan graph has to be interpreted. In India, there is only one. See, India, you should see the India, which is very important. India pattern. Now, COVID. If you see the India graph, it is now coming back and fixed around 20,000 cases. So the number of cases after September are only declining and not increasing. Are only declining and not increasing would mean that either India has gone towards a herd immunity or the new virus has not come or the new virus has not affected. So my idea here will be either we are at the verge of seeing a second peak of the new virus or we have attained a stage of herd immunity. Because in India, guesstimate is 60 crore people have already developed antibodies. That's the guesstimate. Official figures quote it to be between 30 and 40 crore people. 30 crore means 300 million. So the official figure says between 300 to 400 million. But guesstimate is that 600 million people have already developed antibodies. In the worst state, in Dharavi in Mumbai, where there was a big cluster, and for the last many months, there has not been they have now reporting 0 cases because they feel antibodies have formed. So therefore, in those countries where small density people like Australia, New Zealand, they have to worry about the new strain. May not be India, Pakistan, and Bangladesh may not have to worry about the new strain, because with the older strain, we have nearly developed a type of herd immunity. Dr. Sajjad. Dr. Sajjad, if anybody wants to speak out, then otherwise we'll close the meeting. Dr. Jamal?
Well, thank you very much for the presentation. Just I have a question. You were just talking about the herd immunity. When you say that mild and moderate cases do not develop any antibody, and so no question of immunity.
No, I have seen even asymptomatic developing very high antibodies. Oh. These are like, it is not an absolute. These are not absolute, but even asymptomatic cases have seen very high level of antibody. It all depends upon your cellular response. But usually it is seen that if you develop that milder infection may not develop antibody. Will not develop antibody. Russell, Mari.
Yes, Dr. Russell, please. Dr. Russell is there?
I think he's having a cup of tea. He's making
No. Sorry.
I thought you have gone to make a cup of tea for all of us.
No, not at all. Sorry, I would be delighted to make a cup of tea for you and all of you, KK. It was, as usual, very, and I'm glad that you try to make it as simple as possible. Understanding this, the genomics is quite complex. Anyhow, we understanding this, what I'm beginning to think is, with these new strains that are new mutant strains, how we still don't know what will happen with the vaccine, isn't it? Dr. Sajjad, is that what? Do you think the vaccine we might see some effect on the vaccine?
The current situation is unclear, but as on today, we are saying yes, the vaccine will work. The antigenicity may change in terms of making antibodies, but the vaccine at the moment is the best option as early as possible. The earlier we take the vaccine, the better it will be.
But one question here, all the data, I mean, right now we have the two, which are emergency or the registration. So we don't really have too much of data more than 2 or 3 months, isn't it? Or am I wrong?
At the moment, vaccines are not going to harm. Theoretically, they are not supposed to harm. The reason being, you need to understand what are you giving in a vaccine. So if I take a virus, if I take a viral secretions from the nose, put it into alcohol, the virus dies. And if a normal person drinks it, that becomes an oral vaccine. You take the virus and kill it and inject it, that becomes an inactivated vaccine. You take the virus and cut it in the lab, take the spike protein out and inject it, that becomes a vaccine. So making a vaccine is half an hour job, but proving it whether it is effective or not is 1 year's job. So at the moment, there is no reason for virus to be of a concern. The reason being, say for example, the data of China and Russia, they have already given millions of people vaccine for the last 3 months. So China has been able to control their disease. Why? There has to be a role of vaccine there. Why there are no cases in China? And let me see the Russia. Russia has the Russian
Sputnik.
graft.
They have already given, I think, 1 billion people.
Russia is still increasing cases.
Oh.
Russia is still increasing their cases. I don't know. Question comes is, should we take the vaccine or not? As on today, see, there are speculations. Now, what the media will say? They will say, because there was a vaccine hesitancy, therefore this news has been flared up about a new variant, so that people rush towards the vaccine. So people will say the pharmaceutical is behind it. Okay. So these types of things will go on. People will say, there will be people who will be saying, I have seen people debating that this is a pharmaceutical vaccine companies thought they will lose billions of dollars. Therefore, they created a havoc of this new strain. The new strains are occurring every time. So what's new in this strain? If this new strain is there since September, why expose it now after December and create a havoc? If this new strain has not caused any havoc from September, October, November, December, why a fear now? I mean, these are like speculations and debates will continue.
Yes, you are right, Mr. President, Dr. Agarwal. Yes, you are right. And there is another speculation that this vaccine will affect for 6 months or 1 year. And the influenza virus, we have to have every year a shot of vaccine for coronavirus. We still don't know, I think.
That's for sure. That's for sure. It's the annual vaccine.
Yes.
Dr. Agarwal, do you have any idea about this? Yeah, this AstraZeneca vaccine is having 70% efficacy. And what about the other 30%? Whether they have any sort of benefit from vaccinating themselves?
We don't know. We have no data.
I heard that these persons actually, they are not going to have the severe form of the COVID-19 or critical.
Don't know.
They will not go to the.
We don't know. See, as on today, my only concern about the AstraZeneca vaccine is that AstraZeneca vaccine is produced in UK, and till today UK has not given it clearance. That's my disturb. I'm worried about it. Oxford vaccine was picked up by USA and halted twice, and USA gave a clearance much later after halting the trial, and now UK is sitting on the file. Why? Why the UK is not clearing?
But UK has used the Pfizer one. They've already started giving.
Yeah.
I'm trying to say, is it again? Again, people will say it's political because, see, unfortunately, politics plays a lot of role in the. But
Yes,
I still feel is very clearly, if a country adopts a vaccine, if the vaccine is available, if the Pfizer vaccine is available in your country, go ahead. As on today, that's the only vaccine approved. If an activated vaccine comes, blindly take it.
What about the Pfizer vaccine in India? Pfizer vaccine, they have given approval?
Not yet approved. But if in any country an inactivated vaccine comes, blindly take it, because inactivated vaccine cannot harm.
Okay, Dr. Agarwal, can you say that the person who have already infected with the coronavirus, would he take a vaccine even then after having a development? Sorry, after 3 months.
Before a vaccine, you are not supposed to get your antibodies done. You are not supposed to get your antigen test done. You are not supposed to know whether you are asymptomatic, positive or not. Go ahead with the vaccine. And if you have suffered from a known vaccine, wait for 3 months.
But
Okay.
in that case, they are not advising for antibody test. They are telling that anybody can take it.
No.
Pfizer is telling.
Yes, they're saying don't go for the antibody test. So let's close the meeting.
Okay.
Final
Thank-
statement by Dr. Sajjad.
Thank you very much, everybody, for participating. This was very important and very informative presentation by our president, Dr. K.K. Agarwal, and we hope that we all learn very much from today's meeting. Again, thank you very much, everybody.
Thank you.
It's a pleasure to knowing.
Thank
It's
you
a
very
pleasure,
much.
Dr.
Thank you
Sajjad,
for
knowing that you are getting well.
Yes.
Really, it's a word.
Wish you well.
Get well
We
soon.
all-
Thank you very much.
Thank you.
Take
Thank you
care.
very much.